Are you gonna record it to like send to us or post or something?
I can do that if you want.
Yeah, that would be helpful.
Okay. Jackson also asked to meet, so I guess we'll wait for him to log in. How are you guys doing?
Doing okay. Doing okay. How are you?
I'm doing all right. I feel tired for whatever reason today. I didn't think I slept well last night. I'm just pulling up the paper so that I have it handy.
Jack said that Zoom is loading, but he's trying to get in.
Okay, cool. And there he is. All right, what questions do you have?
So for me at least, this just it feels like a lot of things are going on, and I guess I just don't quite understand why it's all going on.
Okay.
I like understand that there's, the two different species. They're testing um, both types of females with both types of males with the tufts and then without the tufts, and seeing just like how receptive they are or aren't.
Mm-hmm.
Um, but first, I guess, um, do they use um, the uh, Rovneri? Is that how you say it?
Rovneri, it's named after a guy named Jerry Rovner, and it's a species that's named after him, so it's Rovneri.
Yeah, so the S. rovneri, do they use that specifically to see if um, if it's pre-existing bias? Like because their species don't have the tufts, they're seeing if they react to it, then it must be a novel trait. Like, is that the point?
Yeah, so I think the thing that will help you is arriving at what the big picture question is in the paper. And the big picture question that George Utz's lab has had for many years is, why do these males have these large conspicuous um, tufts on their legs? And the kind of proximate answer is that females find it attractive, and so if you have large tufts, you're more likely to get a mate than if you don't have them. But the ultimate question is kind of, how did these things come into being? How did they evolve? One idea is that these types of secondary sexual characteristics that females kind of clue into evolve in males as a result of female choice. Females choosing males that have the trait versus don't have the trait. Well, the easiest way for that to come about is for the females to have already been ready to receive that signal once the signal appeared. So the very first spider that had just a little bit of a modification to its legs that made the legs darker or slightly tufted or whatever, that would be a characteristic that would be preferred by females, which would then drive selection on males. So the big picture question that they have here is, can this pre-existing sensory bias for tufted males have been present before tuftedness evolved in males, and as a result of that drove the evolution of tuftedness? So that's the big picture question. Now, the reason that Rovneri is a good candidate for this particular study is that when you look at the figure that has the phylogeny, you see that Rovneri is ancestral to all of these tufted species. So if Rovneri or Stridulans or species A have females that prefer tufted spiders, that would be evidence for a pre-existing sensory bias for tuftedness. You can't really test this in Floridana because Floridana is in a clade where there are other tufted species. Does that make sense?
Mm.
Rovneri don't have the signaling, do they? They don't like move their, like,
They don't have the
other than the
legs.
they don't have the waving signal, right?
I think they do.
Okay, maybe I
think all
because at one point the results say that the males that were tufted but didn't have the signaling whatever of their legs, or using their legs to signal, that was less receptive than those that had a signal and had tufts. And then they go on to say that the least receptive, or the ones that were received least, were no tuft and no signal. So I didn't know how they were controlling for no signal, or how they were causing there to be no signal, unless the Rovneri didn't already have a signal or something.
Yeah. So if you go to page 168 and read the description of the two species.
Okay.
Who was saying something? Was that Kennedy?
Yeah, which page did you say?
168.
Oh.
So these two species are geographically sympatric, but generally occupy different microhabitats. They're virtually identical with respect to body size, genital characteristics, and color. One distinct morphological difference between the two species is the presence of tuft or bristles on the first pair of legs in Ocreata males. During courtship, males of both species produce species-specific substratum-coupled stridulation. This is basically them wrapping on the substrate with their legs and with their pedipalps.
But the next sentence says, in addition, S. ocreata males perform a series of leg-waving displays using the first pair of legs, which contain the conspicuous tufts, but
Rovneri lacks these, yes. There it is, the thing that Jack was talking about.
So that's what it's referring to.
So
they're still tapping, they're just not waving their legs, or Rovneri don't wave their legs.
Rovneri don't wave their legs. So when you look at the video recordings, what they did was they videoed a male doing its display, a male Ocreata doing its display. They videotaped male Rovneri doing their display, and then they digitally altered these videos to either remove the tufts or to add tufts where tufts weren't there before, or to make the tufts larger. Mm-hmm. And then played those back to females to see then what the response of the females is.
Yeah, that makes sense. I was getting lost, the distinction between tapping versus waving legs, because I read that, like,
So if you go to type in Schizocosa into your web browser, and then George Ewitz's last name, and meeting. Let's see if that will get us there. And then click on videos. And one of the things that you'll see is a Schizocosa ocreata courtship video on YouTube. And when you click on that, you'll see a male Schizocosa ocreata basically waving its legs, but you'll also see that, let me share my screen with you real quick here. So here's my search page. This video here, you can see that there is a spider on a leaf. And when you click on that, I don't think you guys can actually hear the audio coming from the video, but they have a microphone that is attached to the container that the spider is in. And you can actually hear the noises that the spider is making as it's going around, moving its legs and also moving its pedipalps. The pedipalps are the male copulatory structures that are right here at the front of the head. And you can see that they're all touching down on the ground. So the legs are waving to either side, but then there are these pedipalps that are touching in the ground, and they use those to create noise as well. And so the way you can remove that, feature of spider courtship is by basically building an arena where the female is on the circular pad, and then there's a space, there's a plastic barrier around this, and then there's another space that goes around this thing, and there's a plastic edge to that as well, and the Walkman is there, so that there's no physical connection between these things, so that any vibration that might be being projected by the TV camera doesn't get picked up as vibrations of the female. So you can either have purely visual stimuli to the female, or you can have a combination of visual stimuli and this vibratory stimuli to the female.
And they removed the vibratory stimuli by placing it on foam or something, right?
Yep. So there's
so
foam installation between these. So the cue that the females are getting is a strictly visual cue.
Okay.
Yep.
So they have a lot of methods.
Yes.
And so they have, the first thing is that they test for habituation and priming. What is that for?
So one of the things that happens when you're going to be testing the same individual multiple times, what can happen is that the individual gets used to whatever it is that you're doing to them, and so stops responding. That's habituation. So
basically, like if they were to realize that it was just a video recording and not actually a live spider, so they'd stop responding.
Yeah,
that's a spider.
Yeah, that's basically a video recording, so I'm not going to waste my time responding to it because there's nothing in that for me. The other thing that happens when you repeatedly expose individuals to stimuli is they can become more sensitive to the stimuli rather than less sensitive to the stimuli. And so what you generally want to do is you would want to randomize the order in which you give stimuli to the individuals so that they don't get used to what's coming. And then the other thing that you want to do is give some individuals the same stimulus over and over again to see if they either start to ignore it or become more sensitive to it. So you want to actually, when you can, you want to actually test for either habituation or priming. Priming is when you become more sensitive. Habituation is when you become less sensitive or less responsive.
Okay, so then why did they test it with live males also?
So where's the live male section?
It's right below on that page.
Oh yeah, there it is. Okay.
Yeah, I was wondering why they didn't do this for S. ravneri as well. They only did it for S. ocreata.
Yeah, and then they proceed to not even talk about it in the results.
No, they did a little bit, I think. They said that but it's the part where there's like something about 90% and 100% or something like that, and then 42% for the smaller size tufts.
On page 173, in the right column, like super in the paragraph right above that new starting paragraph at the bottom. The paragraph starts with female receptivity in response to live males, blah, blah, blah.
Oh, yeah, there it is. Okay. Da, 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 da. Yeah, and so in this case, they're just looking at natural variation in tuft size because they can't remove the tufts from actual animals, and it basically just it shows something different than what the video images show, because in the video images, when we look at the proportion of, well, let's see, let's go back. When we look at the video images, female Ochreata responding to video images, there's no difference due to tuft size. Regular tuft size versus no tufts versus enhanced tufts are all not different from one another. The same thing when we look at the receptivity score, tuft size doesn't make any difference. Regular tufts versus no tufts versus enhanced tufts, those are all not significantly different from one another. And so there is a difference between the responsiveness of females responding to live males versus females responding to video males, but it's mainly in the degree of tuftedness within Ochreata.
Mm-hmm.
It is a weird thing to look at with live males, because you could ask, well, why didn't they also do, because a Ravnarai responding to Ochreata males, or female Ravnarai responding to Ochreata males and female Ravnarai responding to other Ravnarai. Well, for one thing, Ravnarai doesn't have tufts, so there's really no variation there in tuftedness. But what's the other problem with, for example, putting Ravnarai males with Ochreata females, or Ravnarai females with Ochreata males, for that matter?
They're like hetero-specific courtship or whatever.
And what happens when a female spider gets a signal from a male spider that says, "Hey, I'm not your species"?
They attack it.
Yeah.
They kill them and eat them. Yeah. And so that's possibly the other reason that they didn't do a lot of live trials, and especially not the battery of live combinations that you see in the video presentations.
Could they not have, like, I don't really know what working with spiders is like, and I don't like spiders, so I don't want to know, but like, could they just like knock out the spiders somehow and give them artificial tufts on live S. ravnarai?
Oh, these are really small spiders, and they're very difficult. It would be very difficult to remove the tufts from Ochreata, or it would be almost impossible to add tufts to Ravnarai.
Gotcha. Okay.
So yeah, it's just too small and difficult. Now, one of the things that they have done in the past is they have knocked out females to see if the species are reproductively compatible with one another. And they've actually done interspecific mating trials where they'll mate a male Ravnarai to a female Ochreata. And a couple of things happen. The morphology of the legs is different, such that they tend to not have tuftedness, or if the tufts are there, they're much smaller than they are in Ochreata. But the other thing that happens is the courtship mating dance of the males is very messed up. And if you then present those hybrid offspring back to a member of either of the two species, neither of the two parental species recognize the courtship displays of the hybrid offspring.
Not very evolutionarily fit.
Well, yeah, the hybrids are not, and so This is a situation where we have what they call cryptic species, these species that look a lot alike, but they stay separated genetically because of behavioral means. Males and females recognize one another strictly on the basis of behavior. What other questions do you have?
What do they mean by, so what do they mean by latency of receptivity?
Latency of receptivity is basically just how long it takes for a female to respond and orient. So let's go back to my earlier thing here, not news. Here's a video of males versus males. And so you see that this male on the screen is displaying. And if you walk back to the beginning, the question of latency is how long does it take of this male displaying for this male to respond to it? And so here's a male on this screen. Can you see the left-hand screen here? He's in the lower right-hand corner of it. And so latency would be, okay, we started the video playing. How long does it take this dude to walk over there and engage in it? And you see he's already displaying at this male who's displaying. So latency in this case was really, really quick. But in some cases, if this spider is making the wrong signal, the female may just ignore that individual altogether, in which case latency would be longer. So latency is a pretty crude measure of interest. I was talking to somebody, oh, maybe I was talking to you guys yesterday. Who was I talking to? Anyway, think of latency as you're at a cocktail party, you catch somebody's eye across the room, they catch your eye, and then they ignore you versus they catch your eye and then start walking towards you. So that walking towards you is a measure of interest. Now, unfortunately, it may not be an interest in you because that individual you might be starring in a romantic comedy and you catch that person's eye and they catch your eye and they're like, hey, how's it going? And then they walk right past you to the person behind you. But so latency is essentially how long does it take to attract the interest of the other individual.
They didn't find any like significant difference in latency, did they?
Yeah, and it's a crude measure of interest because simply because you walk towards something doesn't mean that you're actually interested in that stimulus. It may just be that, oh, there's something moving over there. And so you orient towards that movement because it's movement, not because you're interested in that movement.
And so, like when they just go through results, for example, the S. ocreata response, they do it for both, but just for example, they talk about, you know, a high proportion of females showed sexual receptivity to S. ocreata controlled males, blah, 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 blah. They go through all this. They talk about a trend. And then they go on and talk about the level of receptivity of female S. ocreata on the basis of scores. And so I guess I don't understand the difference between all those results. Like the first set of paragraph versus the second.
Okay, I can help you with that. It's useful, so look at figure 1 here. It is useful in figure 1 to make sure that you walk through what this is. The top data are ocreata females responding to these stimuli. The second one is Ravneri females responding to these stimuli. But this is in the proportion of receptive females. So if they had 50 spiders, how many of them express some level of receptivity to these stimuli? Figure 2 is similarly structured, female Ocreata responding to these stimuli, female Ravneri responding to these stimuli, but this is the average receptivity score. And that receptivity score is based on table 1. So go back to the beginning here, where you have table 1, where there are behavior patterns that represent a response to male courtship. So there's no response. Lunging at a male basically signals that they want to eat the male. And as a result, that wouldn't be seen as receptivity. Tandem leg raise, raising both legs in tandem, is once again an aggressive display. That's not seen as receptivity. Orienting towards the male without attacking it or making an aggressive display is a level 1 of receptivity. Approaching the male, once again in a non-aggressive way, is seen as some level of receptivity. And then following the male around, once again without attacking the male, is a level of receptivity. But that's not nearly as receptive as walking up to the image and trying to touch the image, or doing what they call a stagger walk, which is kind of this halting walk that they use in approaching the male. And then a slow turn, settling the body to the ground. So normally spiders are kind of up on the ground and alert like this. Settling is what you call it when they kind of flatten their body to the ground. When their body is flattened to the ground, it's very difficult for them to move very quickly. And then leg extending, and especially lowering the cephalothorax or raising the abdomen. The copulatory structures of the female are underneath the abdomen. So if you lower your cephalothorax and raise your abdomen, you're basically giving the male access to have sex with you. And so they have these different levels of receptivity. And the second figure is basically what's the average receptivity score on this numerical rating that they have for how receptive females are. So figure 1 is just proportion of females that got anywhere down the receptivity pathway. This is then how far down that path did you go relative to these different stimuli.
Gotcha.
Does that make sense?
Yeah, it makes a lot more sense.
Could you maybe?
These figures are complicated, so it's important to take them panel by panel. Ocreata relative to these stimuli, Ravneri relative to these stimuli, and are you talking about proportion of females responding versus the degree of response that they had? Go ahead.
Could you maybe explain table 4 and or table 5? I'm kind of confused with, like, I'm not really getting a whole lot. I don't know if I need to get a whole lot from those tables,
yeah.
But
my advice to you is to not pay attention to those tables. My advice would be to pay attention to the figures. Now, okay. There's also something confusing in the figures, in that figure 2, the lower panel, I think has a typographic error in it. That C that you see there should not be there. And yeah, I can't, yeah, it doesn't make any sense there. And so I would pay attention to the figures. The letters above the bars indicate if it's the same letter that things are not different. So here, when we look at mean score, Ocreata responded less well to Ravneri controls than they did to any Ocreata stimuli, but they didn't respond differently to Ravneri with tufts. And also, Ravneri with tufts was not different from Ravneri controls, and both of these were not different from a blank screen. So you could have just as easily been showing them a blank screen. So it. What's that?
You have an awful way to, like, signify differences.
Oh, using the letters?
Yeah.
It's better than, because there are so many different comparisons, it's better than putting brackets above everything.
Yeah.
So things that have the same letter are not different. Things that have different letters are different, which makes this C weird, because if anything, this is not different from a blank screen. This is different from A. B is not different. The Ravneri control is not different from the no-tufted Ocreata male signal. But then, what is C different from? If anything has a C associated with it, it should be the blank screen. So that C doesn't actually make any logical sense over that particular control stimulus.
Okay, so then the next part that kind of like is supposed to tie all this together, I don't really understand all the stuff about like the phylogeny and then how they relate, how these results are supposed to relate to the phylogeny and how they're making the conclusions that they are based on those two things together.
Yeah. So the purpose of the phylogeny, and keep in mind that the phylogeny only has 6 species in it, 6 species of Schizococcus ocreata.
Yeah,
So
that it's like not, you're supposed to take it with a large grain of salt. So
I would definitely take it with a large grain of salt, and I'll show you why here in a little while. So you have a phylogeny, you have these species that are evolving over time. This is the out group. It helps you polarize your characters to show you which direction character chain is evolving. And then you have this out group, this other group of Schizococcus species that have no tufts. And Ravneri is in this group. And then you have a group of species down here that are Floridana, Crassipes, and Ocreata.
Ravneri are ancestral, so they came first, right?
Yeah. So in this case, Ravneri came off of this common ancestor. So this common ancestor gave rise to species A, Stridulans, Ravneri, and the clade that represents all of these guys. Now, Floridana has no tufts, and Crassipes and Ocreata do have tufts. So for a pre-existing bias to occur, somebody back here, earlier in evolutionary time, has to have a preference for tufts. If the preference for tufts evolved here, then that's evolving at the same time that you have tufts arising. And so that would not be a pre-existing bias. You would have the preference for tufts arising back here. And if Ocreata prefers tufts and Ravneri prefers tufts, the two species have the preference for tufts because the common ancestor of those two species had it. If Ravneri doesn't have a preference for tufts and Ocreata does, then the common ancestor could be assumed to not have the preference for tufts, and as a result, you don't have a pre-existing bias. Does that make sense?
So in order for a pre-existing bias to occur, someone back there has to have a preference for tufts, and that's just it?
Yes, that is just it, which is why they tested Ravneri. Because one, they could come up with Ravneri in decent numbers, and two, it represents a basal species in this particular phylogeny.
What does a basal species mean necessarily?
Ancestral. It occurs closer to the base of the tree. So the y-axis here is phylogenetic divergence. And the x-axis is time from the past to the present. So Ravnarai evolved on this branch earlier than Ocreata or Crassipes or Floridana.
Okay, but don't they make the statement somewhere that they can't say it's pre-existing bias?
So here's the tricky thing about figure A and B. In figure A and B, you have two equally parsimonious ways that this has arisen. You could have the independent evolution of tuftedness in Crassipes and Ocreata. That's two evolutionary events. The development of tufts in Crassipes and the development of tufts in Ocreata. Now, an equally parsimonious explanation is that you actually had the evolution of tufts back here, such that this is all tuftedness, this is all tuftedness. That's one evolutionary event, the arisal of tuftedness, coupled with a reversal of tuftedness in Floridana, which represents a second evolutionary event. Therefore, this explanation also is two evolutionary events. And when you do phylogenetic systematics, you basically make decisions on what the correct explanation is on the basis of how parsimonious the explanation is. And in this case, parsimony refers to what's the fewest number of steps that you have to go through to get to a particular result. And in both of these cases, the two explanations are equally parsimonious because both of them only require two evolutionary events to get you there. If this is the case, it's impossible to tell where in this evolutionary line here the preference might have evolved and where in here the tuftedness might have evolved. It could be that the tuftedness was present in the ancestor, the common ancestor, or it might have evolved, this far down on this line, such that tuftedness evolved here, or tuftedness evolved here, or tuftedness was here. If it occurred here or here, then Ravnarai would be a pre-existing bias. But if it occurred back here, Ravnarai might just have gotten its preference for tufts because the common ancestor of the two groups was tufted. Does that make sense?
Better.
Okay. What else?
Did they ever conclude that it was a pre-existing bias? Because in the discussion, they offered like several ways, like reasons why. Like they talked about signal like efficiency or something, and then female choice, and then pre-existing bias. But they said, I don't know, like I didn't really think that there was like a clear conclusion from it.
Yeah, so on page 178, have tufts evolved via pre-existing bias? The last paragraph there, the preliminary phylogenetic analysis provided here suggests that tufts are a derived character in the Ocreata clade that has either evolved once and been secondarily lost in Floridana, or has evolved independently twice in Ocreata and Crassipes. In either case, Ravnarai is in a position basal to the species with tufts. This phylogeny must be interpreted with caution because it is a strict consensus tree derived from 5 equally parsimonious trees. But in all of these equally parsimonious trees, Ravnarai is basal to Ocreata. Since female Ravnarai prefer males with tufts, the female preference for tufts appears to be the ancestral state. It is uncertain, however, whether female preference has evolved because tufts, 1, are novel, 2, they stimulate more photoreceptors and therefore are perceived as a stronger stimulus, and or 3, stimulate a specific pre-existing bias for tufts. So they're basically on the fence about this because they don't have a very well resolved phylogeny.
Okay.
Is it still worth Like, is that one of the important parts that we should be including, even if they're not sure about it?
You should include their conclusion that they are not sure about it.
Okay.
You should include their conclusion that they do not conclude.
All right.
What else?
I think I'm pretty good on my understanding now. I just got to figure out what to like pick and choose to be able to fit into 2 pages.
Okay. What about the other 2 of you?
Kind of the same. There's just a lot of methods and results to draw in.
Keep in mind what the fundamental big question is. Did these tufts evolve because of a pre-existing bias?
Okay.
Let that question help you determine what level of detail to include or not, because that really is their overriding question.
Okay.
So we should focus.
You can go.
So
Go ahead, Claire.
like in our introduction, I focused kind of on explaining the bias and stuff.
Mm-hmm.
Would that be okay?
Yeah. It's necessary to understand what they mean by pre-existing bias and why that's important in sexual selection. Sure.
For future research ideas or like furthering of this research, I mean, I have a couple ideas. I just don't know if they're like, if there's like a glaringly obvious, like this should be improved this way or what, or like this is how it would make sense to further it.
What's the biggest glaring obvious thing that should be done next? And they point to it in their discussion.
I mean, they say a lot of things that could be like done in the future, but they basically give like an example. I think maybe this is wrong, but it says at the bottom left of 178, basically talking about like female choice. Further testing is needed before any strong conclusions are drawn regarding the importance of cues in female choice. And then they give an example. To me, I think they need to figure out the phylogeny thing, making that like more accurate, because that's basically why they can't make any conclusions, is because they don't trust their phylogeny. But I don't know how to do that.
Okay. Look in the acknowledgments. Take a minute here for just a second and read the acknowledgments all the way through. Is there anything in the acknowledgments that gives you any hints about that aspect?
They've got a lot of different moving parts.
They do, but in reference to Kennedy's point.
Kennedy's point was about female choice, right?
No, Kennedy's point was about phylogeny, wasn't it?
Yeah, I said both, but
Is it something to do with like the anonymous person?
No. Anonymous people aren't helpful.
Okay.
Who constructed the character matrix for the phylogeny that they have in this paper?
G. Stratton.
Gail Stratton. Gail Stratton at Ole Miss, University of Mississippi. Type into your web browser Gail Stratton phylogeny.
Should we go to images or like click on a paper?
You should probably click on a paper. Like this paper. Well, let's go back to one that will actually have the figure. Go back. Evolution of ornamentation and courtship behavior in Schizocosa. Insights from a phylogeny based on morphology. 2005, Journal of Arachnology. 2005, Journal of Arachnology. Evolution of ornamentation and courtship behavior in Schizocosa. Insights from a phylogeny based on morphology. How many species of Schizocosa are there?
31.
There are 31. How many did McClintock and Ewitz have in their paper?
6.
2.
7.
In the phylogeny.
6 in the phylogeny. So scroll through this paper showing you the different characteristics that they were looking at, that Stratton was looking at. You can see what the variation in some of the legs are. But get to figure 34 on page 365. What you will see here, let me back it out a little bit if I can.
Are you sharing your screen?
It is
What's that?
shared.
I am
Change
sharing my
your
screen.
change your icon view, Kennedy, or your thumbnail view.
Oh, okay.
So can you see it now, Kennedy?
Yeah.
So here is the phylogeny. You have all these Schizocosa. You have Gladycosa pulchra and Rabidosa ravida are outgroups, but everything else is Schizocosa. And when you get to this place right here, this is showing you tibial bristles. There are tibial bristles absent or present or equivocal. And so here, it means that it's unclear from the phylogeny whether it has it or doesn't.
Gotcha.
So here is the eukaryotic clade. Who all is in the eukaryotic clade?
Rovneri.
Rovneri is deeply embedded in the eukaryotic clade in this analysis. Well, what's the difference between this analysis and the other analysis? Well, this analysis has 49 characters from 31 species. The other one had like 9 characters from 6 species. In this case, is the preference of Rovneri for tufted males at all surprising to you?
No.
No, you should expect them to have that. Why? Because they are derived from a common ancestor that had tufts in an entire clade of common ancestors that had tufts. So Kennedy, you said that the whole female choice thing was important, and also that it would be better to have a good phylogeny, a better phylogeny. Which strategy would you take now?
Phylogeny. Well, I don't understand
Yeah.
if the person like it guided them and they had this information, how they ended up with the phylogeny that they do.
Well, she didn't. This was published in 2005. This is a 2005 paper.
When was ours published? I don't know.
1996.
Ha.
So Kennedy, you are the only one who had the good idea that maybe what we need is a better phylogeny for this group. Gail Stratton did also, and she eventually went and did it.
Dang, Kennedy, that means you like basically have a PhD now.
Basically.
Not quite.
I'll go. Okay.
What else?
I think that's it for me. I do. When they're done, I have a question about my lab report. They're fine to hang around for that if they want to as well, or if they have questions on that too.
I'll go ahead and hang. Might as well.
All right.
Corey, do
Well,
you have any other questions for the Litsum?
No, I think I'm good.
okay. All right. What about the lab report?
Okay, so I emailed you and you said, okay, so then what did you do with those figures?
Yeah, that's exactly what I said.
And so, are you like referencing the figures that we like after they had been produced?
So you created these figures that showed what your data looked like with this best fit line through it. Then, like, what was your purpose in doing that? How did you go about interpreting those? Of what use was making those figures to you in exploring these different functional response types?
Well, we compared them to a typical type 1, 2, and 3 functional response to get a comparison.
Yeah. So put that in your methods also. So you took me as far as we made these things, but then you didn't tell me what you did with them.
Okay. Gotcha.
What else?
I think that's all I had.
All right. What about the rest of you?
Would you mind if I sent you my introduction?
You may do that
Yeah.
with some focused questions on what you want out of it.
Okay, cool.
Luke has sent me his as well, so I have quite a lot of my day today is spent with reading a lot of stuff.
That's okay.
But I'll get around to it and I'll get it sent back to you.
Yeah, I just know that like I feel like the introduction is always one of my weaker points. So.
Mm-hmm. All right. Anything else?
I think I have one more question. Sorry about the Litsum. So if we were to like so for the introduction, I kind of like to break down each component and like work, like you said, work from big overarching and get down to more narrow, so like an inverted triangle,
sure,
or upside down triangle. Would you say the like vague or the largest overarching theme for this Litsum would be female choice and signaling?
I would say that the biggest overarching theme is the general idea of sexual selection.
Okay.
So sexual selection is a process by which two things evolve within a species. One thing that evolves within a species is females choosing males, and another thing that evolves is males competing with one another over females. This whole male competition versus female choice. One of the consequences of female choice is that when females exercise choice, they may lead to selection acting on males, such that males develop over evolutionary time elaborate secondary sexual characteristics, such as bristles on the legs of spiders. Now, that female choice can come about due to pre-existing bias or not. If there is a pre-existing bias, the females are basically just sitting there evolutionarily, waiting for some novel trait to arise in males that they then have a bias towards favoring. And this has been seen as one of the easiest ways in which you can get the elaboration of male traits kicked off. And so these guys are wanting to see if that is maybe responsible for this elaboration of the male trait of tibial bristles in this particular group of spiders.
Okay. Because in my intro, I kind of start with survival and reproduction, and then I talk about how they can be intermingled for spiders, and then I kind of go a little more into like signal communication and female choice.
Yeah, so if you're talking about survival versus reproduction in males and why correct signaling can influence those two things, then sure, I could see you going that direction.
Okay. Cool. Yeah, that's kind of where I went. Okay. I just kind of wanted to make sure I wasn't off track with that.
Sure. No, that would work just as easily as the framework that I laid out.
Okay. Alrighty, I think that's all I have.
Cool. Yeah. Would you mind posting or sending the recording, please? And thank you.
I'll just post this on the YouTube channel,
Okay,
like
great.
all the rest. Yep.
Alrighty.
All right.
Thanks, Dr. K.
Thank
See
you.
you guys.
All right.
Thank you.
I'll see you guys later.